Light energy is transduced into electrical signals by photoreceptor cells in the retina. How processing through layers in the retina creates center surround receptive fields for retinal ganglion cells. We talked about how visual information is organized and channeled through different layers in the lateral geniculate nucleus of the thalamus, through different layers and the blob system of V1, and through different stripes in V2, the thick and thin and interstripe regions. And then at the end, I mentioned how information from V1 and V2 is sent to a hierarchy of other visual areas, broadly divided into the ventral pathway, the what pathway that processes information about what is out there in the world, and the dorsal pathway, which processes information about where things are in the world and how they're moving through the world. This diagram here just shows a few of the major, most studied areas in the ventral and dorsal pathway, but in fact, the number of distinct visual areas that have been identified in monkeys, and there always turn out to be homologs in human cortex, is on the order of 40 now. And it's a reasonable question why we need so many different visual processing areas, why we need so many processing steps or stages beyond primary visual cortex, or V1. Why is it that we can't just use image information as it comes from the eyes? Well, images are just huge arrays of numbers, like the values for the pixels in a camera, or the activation levels of rods and cones in the retina. And if I gave you an array of numbers that corresponded to a camera display of this room, you'd never be able to figure out what was in the image just by looking at the numbers. And you could try to write a computer program that would do it, but people have been trying to do that for over 40 years with limited success. So the way you would do it is to cheat. You would use the numbers to print out an image, and then you'd use the algorithms in your brain to achieve that. So why aren't images any good? Well, they contain all the visual information we're going to get, certainly, but that information is not in a useful form. First of all, it's too distributed across millions of channels. It's a huge signal, requires a lot of processing, too big a signal to send over axons to other parts of the brain because it would take up too much of your axonal bandwidth. Too big to store in memory because you'd have to take up too much storage space in terms of reweighting synapses. The signal is also way too variable. So as I move my eyes around in the room, or I move around in the room, or other things move, or lighting conditions change, the actual signal on my retina changes completely. And that wouldn't be very useful for storing in memory or sending to other parts of the brain, because you'd have to be able to recognize a virtual infinity of things. You'd have to be able to store a virtual infinity of things just to correspond to one thing that you wanted to know about the world. For example, the face of a friend. Finally, the information about stuff in the world that we care about in an image is extremely implicit. It's all there, but it's essentially hidden. It would take many operations, and it does take many operations through higher level visual cortex to pull that information out. Once visual cortex makes that information explicit, then it can actually be interpreted easily and quickly by the rest of the brain. So what visual cortex has to do is make implicit information explicit, to make variable information more stable, so you only have to store the same thing in memory and you're sending the same signal to other parts of the brain. And it has to vastly compress the signal, so instead of requiring millions of channels, it can require a reasonable number of channels for sending signals to other parts of the brain. So the ventral pathway is processing images to produce compressed, explicit, stable information about what is in the world. The dorsal pathway processes images to produce compressed, explicit, stable information about where things are in the world and how they're moving. So as far as compression, we've already talked about the first major compression step in vision in the retina, where redundant information about regions of constant luminance or color is largely filtered out, and the center surround receptive field structures of retinal ganglion cells preserve information about contrast regions, about borders between different luminances and different colors. So that's a major compression step. So now we'll talk in part about how the ventral pathway further compresses information about shape on the way to an explicit, compressed representation of structure of things in the world. So this is a picture of the monkey brain, right hemisphere seen from the side, right hemisphere seen from below. This is a diagram of many sub-areas in the ventral pathway, which you don't have to know the details of. Monkeys are the animal model that is the source of most of what we know about high-level visual processing, because we can't do experiments with really high spatial or temporal resolution in humans, but everything we've seen so far suggests that visual function in higher primates, including monkeys and humans, and also the precise organization of these 40 or more areas is remarkably conserved across species. So we'll start out talking about processing in primary visual cortex, or V1, which we know in humans and also in monkeys is in the back of the brain at the occipital pole. So compared to retinal ganglion cell receptive fields, which we've already learned about, V1 receptive fields are slightly larger. So V1 receptive fields near the fovea subtend fractions of a degree of visual angle. A degree of visual angle is subtended by your pinky held at arm's length. So a V1 receptive field is going to be a piece of that width. So on that scale, in our world, if you look at slices of the world, the size of a V1 receptive field, what you see isn't just random pixel patterns. We don't live in a random world. On this scale, in the natural world, luminance and color borders tend to be extended edges. And that's because in our natural world, things, objects, surfaces have a high degree of smoothness on this scale. We don't live in a world of constantly really spiky things because of physics, the properties of different materials, biological growth processes, construction practices. Most of the things in our world are smooth like this on the scale of V1 receptive fields. And that's what explains the most salient aspect of processing in V1, orientation tuning. Orientation tuning in V1 was discovered by Hubel and Wiesel in the 60s. David Hubel and Torsten Wiesel got the Nobel Prize in part for this work. So they came from a retinal physiology lab originally, and when they started studying primary visual cortex, or V1, they tried to use the same stimuli that they'd used in the retina. And in the retina, based on center surround receptive fields, we know that you get really strong response if you show a small bright dot or a small dark dot, depending on whether you've got an on or off-center cell. That didn't work very well as stimuli for V1, but what Hubel and Wiesel discovered, as they described serendipitously, was that neurons did respond to edges of contrast moving across the receptive field. And here's one of their early experiments. You can tell how early it is because the data here is actually oscilloscope traces in the original publication. And in this trace, of course, this is time in this direction. All the vertical hash marks are action potentials fired by the V1 neuron that they were studying. And if you spread this oscilloscope trace out in time, each action potential would have the shape that you're familiar with. So here's the stimulus situation. What they've done is to draw the receptive field as a dashed rectangle. They have a dark bar, and they're sweeping that back and forth across the receptive field. And what is clear for this cell and characteristic of most cells in V1 is that the cell was selected selective for a particular edge or bar orientation. So this diagonal bar at 135 degrees swept back and forth across the receptive field evoked lots of spikes. If you went a little bit off in orientation, still a number of spikes, but a weaker response. As you get even further away and towards vertical or horizontal, the response goes away entirely. So why orientation tuning? Well, orientation tuning can compress the information about a chunk of the visual image like this into one signal. What had required a whole line of retinal ganglion cells to represent is now represented by one signal coming from one V1 neuron. And in fact, Hubel and Wiesel's model for how V1 receptive fields were formed was that they got input from a line of retinal ganglion cells which conferred an on-center stripe surrounded by off-centered stripes, or conversely an off-center stripe surrounded by on-center stripes. So this is a major compression step based on the statistics of shape in our natural world from many retinal ganglion cells into a single signal for a local edge. As you progress forward through the ventral pathway, through multiple stages, V2, V4, all of these stages in the inferior part of temporal cortex, receptive fields gradually become larger because at each stage a cell is getting input from lots of cells from the earlier stage, inputs that have different retinal locations for their receptive fields. That sums up to a larger receptive field. So by the time you get to intermediate stage V4, which is here in the monkey brain and has also been identified in the human brain, receptive fields are larger. They cover several degrees of visual angle near the center of gaze, near the fovea. And on this scale, our world, our natural world, is somewhat less smooth. That is, a contrast or color border on this scale is likely to have changes in orientation, not just a single continuous orientation. So if you wanted to compress information about chunks of the visual world this size, you would need to be able to represent those changes in orientation. Change in orientation is what we call curvature. It can be very abrupt, where you have an angle, so that's very sharp curvature. It can be gradual and smooth, like a circle. It can be either convex, something pushing out from the center of the figure, or it can be concave. It can be an indentation into the figure. So it turns out that many cells in V4 do precisely this. That is, they explicitly represent curvature or change in orientation within their receptive fields. And this is an example of an experiment that shows what that looks like. So this is an experiment on a single neuron in area V4, recorded from the brain of a monkey who was looking at little shapes being flashed on a computer monitor. The shapes are the little white icons in each of these circles, and the darkness of the background circle indicates the strength of the response of the cell to each of the shapes, running from no response at all, around 0 spikes per second, to over 30 spikes per second, which would be a very strong signal from the cell. You can see that this neuron responds to lots of different shapes. Many people have thought that there would always be some single best shape that would define the responses of visual cells in higher level cortex. That turns out not to be the case. But all of these high response shapes do share some local structure. They all have some kind of convex, fairly sharp convex curvature near the top of the shape. And you can represent that with a tuning function, which is shown down here. So the x-axis for this tuning function represents position relative to the rest of the shape. 0 means off to the right, 90 means near the top, 180 means towards the left, 270 means towards the bottom. The vertical scale is contour curvature, where 1.0 means very sharp convex, 0.5 would be broader convex curvature, 0 would correspond to a flat contour, negative numbers to concave curvature. And the color scale here represents the tuning of this neuron, which is just what you'd expect from looking at these shapes. Its tuning peak is for sharp convex curvature at an angular position of 90 degrees. That means near the top of the shape. So in addition to compressing information about change of curvature on the scale of V4 receptive fields, these responses also are consistent with something that theorists long proposed about how the brain could achieve compressed, stable, explicit representation of object shapes. The idea of configural or parts-based coding. The idea that entire shape would be represented by the ensemble activity of a number of neurons representing the parts of the shape and their positions relative to the rest of the shape. This is an example from an early theory paper where it's imagined that the human shape would be represented in terms of a cylindrical torso, cylindrical forearm and upper arm, cylindrical legs, a globular head. So a parts-based representation would achieve compression in the sense that if this is your description of the human shape, the number of parts is on the order of 10. That's a much smaller number of signals than the number of photoreceptors needed in the retina, or the number of orientation cells in V1, or even the number of curvature cells in V4. The representation, a configural or parts-based representation, is explicit about structure. We need to know about the structure of things in our world. We do have good cognitive access to the structure of things. We're all capable, for example, of describing the structure of the human body. We need an explicit signal for that kind of structure, and this would provide it. So this is an analysis showing how parts-based or configural representation would work in V4. So we had this example neuron here that basically responded to shapes whenever they had sharp convexity towards the top. So other cells not shown here had tuning peaks at different points in this curvature position space. And what's done here is to take each of those tuning functions and weight them by the response to this shape here. So this is one of the shapes from that experiment. It's a shape that drove a strong response from this cell. So this cell got a strong weight, and its tuning function in the weighted average contributed to this peak here. Other cells tuned for concavities at the upper right or the upper left contributed to these peaks here. Other cells contributed to these two peaks that correspond to the blunter convexities on the right and the left. And this peak here corresponds to the really broad convexity near the bottom of the shape. So the entire shape here is represented in terms of peaks across the V4 population that correspond to the contour sections that we would actually describe if we had to describe this shape. We'd say that there was a pointy bit up at the top, that there were two projections to the right or left, indentations at the upper right and upper left, and a broad shallow bottom. And in fact, this representation is complete enough that you can regenerate the original shape based on this signal with a decent degree of accuracy. So we talked about V1, V4. When we get into higher stages of processing in the inferior part of the temporal lobe, which is also called IT, inferotemporal, receptive fields get even larger, large enough to encompass whole objects. And you might think that at this stage you'd finally see cells that were tuned specifically for a particular shape, like the shape of the dog. But that's not the case. It remains the case that cells are tuned for partial shape, and the representation of a stimulus, like a dog, is still based on the ensemble activity of a number of cells signaling the different parts of the dog and the position that they occupy. But it is true, as you get to higher level stages in the ventral pathway, cells put together inputs from lower levels to generate information about more complex parts, more complex part configurations. And this is an example here. So this is a single neuron in the posterior part of IT, the very next stage after V4. So it's getting inputs from a number of V4 cells. It responds to shapes that have concave curvature facing towards the lower right, the sort of thing that you would see some V4 cells respond to. So this is its response in spikes per second through time after stimulus onset. So the stimulus presentation period here is the gray region. Vertical axis is spikes per second. Horizontal axis is time in milliseconds. So it responds to these concavities facing towards the lower right, but it also responds a little bit to stimuli with concavities facing towards the upper left. But it has a really large and continuous response to stimuli that have both of those concavities. So this cell looks like it's putting together information from multiple kinds of V4 inputs to develop selectivity for this sort of neck-shaped configuration defined by the two opposed concavities. So configural coding is compressed because the number of parts is much smaller than the number of pixels. It's explicit about object structure because it's telling us what type of parts there are and where they are. And another advantage of configural coding actually is stability. So if part position is represented not in eye-centered coordinates, which is the original reference frame for all vision, but instead represented in an object relative reference frame, that is a reference frame where being to the right or above or below the center of the object is what's important, not where you are on the retina, then even if the object changes position on the retina, as it does constantly as you move your eyes around, the representation stays stable. And in fact, as you move forward through the ventral pathway, you go from cells having small receptive fields and being mainly sensitive to eye-centered position to cells having very large receptive fields, thus no longer having sensitivity to eye-centered position, but having really exquisite sensitivity to object-centered position. And that's exemplified here. So this is another cell in PIT. This is a cell that responds to a combination of concavities that define the joint between a torso and a projection or a leg pointing downward. So these two, concavities pointed out in color here. And when that configuration of concavities occurs on the left side of the object, really strong responses peaking around 40 spikes per second. If the exact same configuration occurs to the right of the object, little or no response. So the stimuli in this experiment are small, only covering a few degrees of visual angle. So this is a very small change in absolute or eye-centered position, but it's a big change in object-centered position. It's a big change in what we would describe in words if we were describing a shape like this, whether something was on the left or the right. And this shows how the ventral pathway becomes very sensitive to object relative position, not to eye-centered position, and that's the basis for stability across changes in position and even changes across size. So objects are different sizes on the retina depending on whether we're close or far. An object-based reference frame is one that changes with the size of the object. So we not only need to know about object structure, we need semantic knowledge about object identity. We need to know that this is a dog, for example. Abstract semantic information is not represented in visual cortex. Visual cortex processes structure. But it is generated in prefrontal cortex based on inputs from final stages in the ventral pathway. So final stages in the ventral pathway project up to this region of visual cortex, of prefrontal cortex, sorry. Prefrontal cortex stores visual information in working memory and uses it to make decisions and guide behavior, because prefrontal cortex in turn projects to motor cortex areas, including areas that control language production, allowing us to say the word dog when we see a picture of a dog. This is important. Visual cortex doesn't just process information for the sake of it. The whole point is to send information to other parts of the brain so that it can be used to guide behavior, to inform cognition, to be stored in memory. So these projections from final stages of the ventral pathway to prefrontal cortex are important for using visual information to guide behavior. These parts of the ventral pathway also project to orbitofrontal cortex. That is cortex that's on the underside of the front of the brain here, right above the eyes. And orbitofrontal cortex is involved in processing the value of objects, how much they're worth to us in terms of reward. And the ventral pathway also sends information in this direction into structures in the medial temporal lobe. Entorhinal cortex, perirhinal cortex, and by extension into the hippocampus and the amygdala. So this is how processed information from the ventral pathway, compressed, stable, explicit information about what is in the world, gets into long-term memory so that we can access it later, minutes later, hours later, weeks or years later. So getting back to semantic representation in prefrontal cortex, this is an experiment done by Earl Miller at MIT in monkeys, trained to make an essentially semantic or binary abstract discrimination between dogs and cats. The stimuli were based on actual 3D scans of cats and dogs, and then partially morphed versions of cats and dogs that were 80% cat, 20% dog, 60% cat, 40% dog, etc., creating a whole space of shapes. And the monkey was required to learn this decision boundary here, where everything on one side had to be regarded as a cat, everything on another side had to be regarded as a dog. And here are the responses of a cell in prefrontal cortex during this task. So the stimulus was presented during this period. And then during this delay period, the animal was required to remember whether a dog or a cat had been presented. So it's a task that taxes the working memory aspect of prefrontal processing. And so here's the space of cats and dogs that were presented to the monkey. On this edge of the space right here, you've got 100% dogs. These are 80% dogs. These are 60% dogs. This is the decision boundary. Then you've got 60% cats, 80% cats, 100% cats. So right around the boundary, things look extremely similar. And if you look in visual cortex, their representations will be similar because their structures are similar. But when you look in prefrontal cortex, you see this very distinct separation between the two categories. So 100%, 80%, even 60% dogs for this cell evoked a lot of activity during the delay period, whereas different levels of cat information evoked much less activity. So this is a cell in prefrontal cortex that provides abstract information about the categorical identity of objects that are important in this task. Okay, so to review this part, things to know about ventral pathway object processing. First of all, the goal of all of this processing is to turn image information, which is distributed, variable, and implicit, into useful information for the rest of the brain that's compressed, stable, and explicit. In early stages like V1 and intermediate stages like V4, neurons achieve compression with tuning for geometric elements that are common in the natural world on the scale of their receptive field sizes, edges in V1, curvature fragments in V4. At intermediate and higher level processing stages in V4 and IT, object structure is represented by configural population coding. Individual neurons represent parts or groups of parts. And configural coding is compressed because a few parts can be used to describe a whole object structure. Stable because everything's been transformed into an object-based reference frame instead of an eye-centered reference frame. And explicit about structural information. And then finally, abstract semantic information about things like object categories, like dogs versus cats, is generated in prefrontal cortex based on inputs from higher level ventral pathway cortex. Other important outputs from ventral pathway to orbitofrontal cortex, dealing with reward, to medial temporal lobe structures where object information can be stored in long-term memory. So that's the outline version of what's important to know about ventral pathway processing. Any questions about ventral pathway? Okay, so on to a little bit about the dorsal pathway. So last time we talked about how where information, motion information flows through a specific layer in V1 called 4B, through the thick stripes in V2, and from both V1 and V2 into area MT. MT stands for middle temporal, and MT is one of the first stages in the dorsal pathway. So motion processing actually begins back in V1. So there are many cells in V1, especially in layer 4B, that are sensitive to motion and specific motion directions. So I didn't mention this about this example that we looked at before, but this cell is not only sensitive to orientation, this cell in V1, it's also sensitive to direction of motion. So in this experiment, Hubel and Wiesel were passing the bar back and forth across the receptive field. In this case, when the stimulus passed in this direction, moving towards the upper left, lots of spikes. In this direction, almost nothing. So this cell, in addition to being orientation tuned, is tuned for motion direction. As I said, where information from V1 and V2 projects to the dorsal pathway, beginning with area MT, which is in a sulcus right around here, also projects to areas in the medial wall right here, the medial wall of the parietal lobe. Here's an example of motion direction tuning from MT. Same kind of experiment that Hubel and Wiesel were doing with moving bars in different directions. This is a classic MT cell with really strong responses to motion towards the lower left, some response to motion to the left or downwards, and almost no response in this null direction. So motion tuning even stronger in MT and more common. So almost all cells in MT are tuned for motion. So the dorsal pathway processes motion information. It processes position in large-scale space. Maybe the most important output function of the dorsal pathway is to provide visual information about all of these things to other parts of the brain to guide our actions in space. In fact, for a long time, the dorsal pathway has been called the where pathway. Some people say it should be called the how pathway because it basically uses visual information to guide how we move through space, how we interact with things physically in the world. So major outputs from dorsal pathway to other parts of cortex are represented by different colored arrows in this diagram. We'll talk about the red arrows first. So, well, the red and green arrows. So there's a dorsal pathway area called MIP. It stands for medial intraparietal. You don't have to remember the name, which provides information to dorsal premotor cortex, which controls arm movements. So that's this red arrow going right down here. Dorsal pathway area LIP, very close. Another name you don't have to remember, stands for lateral intraparietal area. Provides visual information to the frontal eye fields. So this is the green arrow system here. Frontal eye fields here are labeled 8A46. The frontal eye fields are part of prefrontal cortex with very direct control over eye movements. So if you make a decision that you're going to move your eye to look at different things in the image voluntarily, that's based most immediately on activity in the frontal eye fields. So dorsal pathway area LIP provides visual information to the frontal eye fields to guide where those saccadic eye movements are going. Here is an experiment that demonstrates the difference in information coming from MIT to guide arm movements and LIP to guide eye movements. This is an experiment in which a monkey was trained to either make eye movements or to make reaching movements to target lights at different positions. And this is an example cell from LIP, which projects to the frontal eye fields. This is an example cell from MIT, which projects to the arm representation in premotor cortex. These are responses during trials in which the monkey was making eye movements to the target and keeping its arm still. These are responses on trials in which the monkey was keeping its eyes still but reaching to the target. When the monkey had to make an eye movement to the target, lots of activity from this cell in LIP. But when the monkey had to make a reach motion to the target, not much response, even though in both cases the visual stimulus was exactly the same. Here's the MIP neuron that projects to premotor cortex. Little or no response during the eye movement trials, but a strong continuous response during the reach trials. Dorsal pathway area VIP and AIP, again, the names don't matter, provide visual information to the hand representation in ventral premotor cortex. So this is the lower red arrow right here, which controls grasping movements. One of the important roles of the dorsal pathway is to transform information between reference frames, and in particular to transform information from the original eye-centered reference frame into reference frames that are relevant for particular tasks. And when it comes to hand-based grasping movements, it's the hand itself that provides the relevant reference frame. And that's demonstrated by visual responses in ventral prefrontal cortex here. This is a cell that has a visual receptive field, but the visual receptive field depends on the position of the hand. So if the hand is way back here, this is the part of visual space the cell responds to. If the hand is here, the cell responds to this part of visual space. If the hand is here, the part of visual space that the cell responds to moves over along with the hand. The dorsal pathway also provides visual information to guide navigation through the world. So that's the set of pathways represented by the blue arrows. This pathway goes through areas of the brain on the medial wall, including posterior cingulate cortex, or PCC, right back here. And eventually, these terminate in parahippocampal cortex, in a part of the brain that in humans is called the parahippocampal place area. So in an fMRI experiment, you can see responses to pictures of landscapes and buildings and room interiors in the parahippocampal place area. So for navigation, for understanding scenes, you want the reference frame actually to be world-centered. You want it to be stable. You want your idea of where things are in the world and how to move toward them to be stable, even if you move your eyes around, even if you point your head in different directions. And this is an experiment, the details aren't important, that demonstrates that cells in posterior cingulate cortex on this blue pathway coming from the dorsal pathway in fact represent position of action targets, in this case targets for eye movements, in a world-centered reference frame that stays stable even if you move the eyes, even if you move the head. If you plot this cell's response field for targets in terms of world-centered coordinates, the response fields line up, even if you're changing eye position or head position. Okay, so things to know about dorsal pathway processing of motion, space, and action. Motion processing begins in V1, continues through MT and higher areas in the dorsal pathway. Dorsal pathway represents eye movement targets and projects that information to the frontal eye fields to help control eye movements. The dorsal pathway represents visual information about arm and hand movements and projects that information into premotor cortex to guide reaching and grasping. The dorsal pathway is important for navigation and scene understanding, and that is supported by its projections into medial wall areas like the posterior cingulate cortex and eventually into parahippocampal cortex. And finally, a major aspect of dorsal pathway processing is to transform visual information from the original eye center coordinates into coordinate systems that are relevant to particular tasks. So that's all. Any questions? Okay.